Thank you so much for the invitation to speak here. Today I'm going to be talking about confounds in neuroimaging, focusing quite a lot on describing the different confounds that we typically encounter in neuroimaging studies and some of the more popular or common approaches for addressing those confounds. And then I'll end the talk with some exciting new machine learning techniques that are being developed to deal with confounds and nuisance variables. Okay, so in my community, we don't really make any distinction between confounds versus nuisance variables. We just call them all confounds. Artifacts are called confounds. Anything that you don't want to happen, that may happen with neuroimaging data, is just called a confound. But technically, a confound is an extraneous variable that affects the neuroimaging or the non-imaging measures and systematically differs across individuals in the sample or in the overall population. A nuisance variable is an extraneous variable that has an association with the neuroimaging and or non-imaging measures, but doesn't contain any pertinent neurobiological information that's relevant to the study question. Random sampling, best practices in data collection, and state-of-the-art data pre-processing can minimize confounds and nuisance effects, but effects do persist even after some of these basic things, and they have to be accounted for in neuroimaging analysis. So just to give you a big overarching picture of neuroimaging studies, and before I introduce confounds, I'm going to focus here on multimodal MRI because that really, MRI data sets typically have the larger sample sizes that are relevant, for machine learning techniques. In this case, you can put a person in the scanner. You might collect several different kinds of scan types, including a structural scan that reflects anatomical information, fMRI data, and these are scans that last maybe five minutes or 10 minutes, where you get snapshots of the brain every second or so, while a person might be doing a task or just engaged in idle thinking. You can also collect diffusion data that allows you to look at white matter microstructure. So for each one of these scan types, we might compute several multimodal brain maps that reflect different aspects of brain structure and function. Different aspects of gray matter morphometry, of functional activation and functional connectivity, and of white matter microstructure. Each of the scan types undergoes extensive pre-processing to address artifacts and other confounding effects in the data that are well known. Structural scans may undergo some intensity normalizations and skull stripping and things like that before you can use them to compute morphometric measures. fMRI data, one of the biggest confounds for fMRI are motion effects. So we pay a lot of attention to motion correction, but there are also other factors that can degrade bold fMRI signals and cause signal dropouts and distortions, and all of these things have to be corrected for in pre-processing. And I'll just add here that there really is a multiverse of pre-processing strategies for pretty much every imaging modality. So this can create a lot of variability in the computed outcomes, and you kind of have to take this into account for bigger data sets that might combine data across studies or multiple sites. It's common to compute a variety of experimental confounds that can be used in subsequent analysis. And I'm just showing an fMRI prep diagram here that's a kind of a standardized tool for data pre-processing, and it will automatically put out some of these things that we'll talk about in a minute. Okay, so brain maps can be computed in a variety of ways for structural MRI, for example. A structural image may undergo segmentation, nonlinear registration, and then that information is used to calculate a gray matter volume map. For fMRI, you take the time series and you do some statistical modeling to calculate a brain activation map or a brain connectivity map. This is usually implemented with a standard general linear model, where each voxel's time course during a task performance is modeled as a linear, just as a regression, where you have a model of the stimulation. In this case, this would be an on-off design that I'm showing in this regressor. And you fit that to the voxel's time series, and then you log the regression weight at that voxel's location. You do this for every location in the brain, and you build up this brain activation map. And so these are then features that are passed to higher level analyses at the group level. And group level studies generally focus on assessing case control comparisons or brain behavior associations via massively univariate or voxel-wise general linear modeling. So for fMRI, this is a second level model. And in this case, you have the brain maps for a particular modality for all the participants, and you extract the subject values at each voxel location, and you may fit a model to them that encodes group membership, and then create a contrast of the regression weights encoding group membership to get a map that encodes the group difference in, say, a task activation. So this has really been the workhorse of fMRI analyses and of structural MRI, comparing gray matter volume between groups and so on. Most neuroimaging studies have small to modest sample sizes, maybe around 50 participants. This is definitely changing over the last few years, but by far the vast majority of the published literature is for relatively small sample size, with small to modest amounts of data collected per subject. Anybody working in neuroimaging has probably heard about the replication crisis that we have from underpowered studies of case control differences or brain behavior associations. So most neuroimaging studies reside down here in terms of, less than an hour's worth of imaging data collected in tens of participants. However, there are now over the last decade, the development of these larger scale neuroimaging data collection initiatives, including the Human Connectome Project and the UK Biobank, that have collected data in more than 1,000 people in the case of the HCP, and ongoing data collection to collect neuroimaging data in 100,000 people for the UK Biobank. Okay, so like I said, in 2010 roughly, the Human Connectome Project started. ADNI, which is an Alzheimer's disease focus neuroimaging initiative, started even before that and has gone through several different iterations. And there are other large-scale repositories, the 1,000 functional connectomes, and large-scale multi-site studies like the UK Biobank and the Human Connectome projects related to health and disease, as well as the Human Connectome projects related to lifespan in healthy individuals. So, with these bigger data sets now really being embraced by the neuroimaging community, everybody wants to use them because, finally you get sample sizes large enough to have well-powered studies. Unfortunately, it brings along some issues with conventional statistical analyses. So, we might have these multiple modalities in each participant, and we may have hundreds to thousands to 100,000 participants worth of data. And typically, for the large-scale neuroimaging initiatives like UK Biobank and HCP, they do a lot of phenotyping to collect a lot of non-imaging data as well. So it's just a big explosion of data. Conventional statistical analyses using that hierarchical GLM approach I described for fMRI, or just GLM for group level analyses, they really are just unimodal analyses, examining, case control differences, a single modality at a time. If you want to link group differences in gray matter volume to group differences in task activation, that's usually done post hoc by the researcher. So, we have to kind of think about maybe using multivariate methods here that can really help solve this data explosion problem. Stringent corrections for multiple tests have to be done, and they have to take into account relatedness. In the HCP young adult, twins and siblings were purposefully recruited, so there's a lot of rich family structure there. In studies like the ABCD, which is a large-scale neuroimaging initiative collecting data in 10,000 kids longitudinally, there's a lot of nesting. So kids were recruited within schools, within sites, and so on. So you have to take all of that structure into account. Phishing and p-hacking become very easy when you have a lot of modalities and a lot of subjects and a lot of non-imaging measures. But confounds actually become a much bigger problem with the more subjects that you have. In the standard null hypothesis testing framework, with the large sample sizes, the good news is that now you're powered to detect those subtle effects that are not detectable with a small sample size. The bad news is that all the subtle effects will be statistically significant, even if they do not explain much variance in the data related to the biological effects of interest. For example, in this paper by Smith and Nichols, they show that with data from almost 15,000 participants from the UK Biobank, even with Bonferroni correction for 14 million association tests between image-derived phenotypes, things like volume of amygdala, connectivity between two brain nodes, between those association tests between those IDPs and non-imaging variables, like tobacco use and cognitive test scores, that statistical significance can be reached with much less than 1% of the variance in the non-imaging variables being explained by the image-derived phenotypes. So, you get high level of significance, even though the relationship, it's not, the variable isn't even explaining 1% of the variability in the non-imaging phenotype. The American Statistical Association has also emphasized that statistical significance is not equivalent to scientific, human, or economic significance, that smaller p-values do not necessarily imply the presence of larger or more important effects. So this just becomes a problem. And because you're sensitive to such subtle effects, you're then exquisitely sensitive to confounds, which can be not so subtle effects. So what do we do? Well, unsupervised learning methods like ICA and other techniques like that can help tame high-dimensional data and potentially leverage cross-modal information and do this data fusion, handle that data fusion problem a little bit. Machine learning for classification and prediction. It becomes very sensible when you have these large data sets, and that is, those are multivariate techniques that can really help too. And so, of course, people have really jumped on the machine learning bandwagon in neuroimaging, and a PubMed search for machine learning and MRI and brain turns up an exponential increase over the last, few years in the application of machine learning for neuroimaging data analysis. If you do prediction and machine learning and MRI and brain, you actually get about triple this. But it turns out about 40% of imaging studies that report prediction are really just looking at associations. So Poldrack reported that in his paper on guidelines for best practices in machine learning of imaging data. Okay, so people are really embracing machine learning techniques. In this case, you may use these different brain maps and derive some brain features or image-derived phenotypes and put them into some kind of analysis where you might want to predict age or disease status or behavioral scores or something like that. An important goal of most brain imaging studies is to gain scientific insight into brain behavior relationships by isolating the specific contributions of brain measures to reveal how behavior may depend on each brain measure. The GLM and inference via the null hypothesis statistical testing framework has been the workhorse to achieve this goal even though there's debate about how significant associations may relate to causal influences. So, these methods can do well to tackle some of those issues. Okay, so a key goal of many studies that leverage machine learning classification or prediction methods is not merely obtaining a model with the highest classification or prediction accuracy. In some cases, that is definitely the goal. But mostly, at least the researchers that I work with, they want explainable models that advance our knowledge of the neurobiological mechanisms underlying clinical and behavioral characteristics. They want models that are neurobiologically plausible and capture important aspects of brain function. They want trustworthy models that can inform clinical decisions, and they want to know how and why the model works to understand when it may fail and to understand its generalizability. Models developed for prediction can provide biological insights. However, for neuroimaging-based machine learning models relating brain structure and function to behavior to be interpretable, they should be comprehensible to humans. They should provide useful information about what mental or behavioral constructs are represented in particular pathways or brain regions. And they need to demonstrate that they are based on relevant neurobiological signal, not on artifacts or confounds. Some models are neurobiologically plausible and capture important aspects of brain function, whereas the ones that capitalize on confounds to achieve high prediction or classification accuracy really just don't contribute to our understanding of the brain. While methods for controlling confounding variables are somewhat developed for classical statistical analyses, and I'm going to talk about those, they're very simple approaches that are actually in pretty widespread use. So I will cover those. Procedures need to be adapted for high-dimensional predictive modeling settings. Failure to adequately address confounds can lead to predictions being driven by confounding effects that affect both the brain imaging data and the prediction targets, but are irrelevant to the problem at hand. And they can make interpretation of brain behavior relationships revealed by predictive models difficult because they can mediate observed associations, or they can be a latent common cause of observations. So now I'm just going to talk about some of the some examples of the confounds that are very common in neuroimaging studies. So we have the confounds at the subject level. The most important one is subject motion. Subject motion will contaminate every MRI modality. No MRI modality is immune from subject movement. It's a huge source of noise and artifacts for all modalities. And here I'm just showing a carpet plot for a functional MRI time series, where each row in the carpet plot is a voxel's time course. And then I'm showing the time courses here of global signal, CSF signal, and so on. And we see our well-behaved participant here, who's laying quite still, has a nice, just white noisy looking carpet plot. This is great. Whereas this participant, we see several discontinuities in the carpet plot, lots of intensity changes, banding in this carpet plot. And these occur at time points where the fractional displacement of the brain in the scanner spikes. So there's a movement there. These outputs are generated from fMRI prep, and these are some real subjects that we have in one of our data sets. So you can get a good picture of how motion might be impacting the data for a particular subject. And you have to do lots of things to try to fix this if you want to keep this person's data set in your study, in your data analyses. Other confounding effects can be physiological motion from cardiac pulsatility, respiration, and low-frequency blood oscillatory signals, scanner effects, and non-imaging equipment like visual stimulation delivery systems and things like that can all have problems, and those can manifest in the data. This would be RF spikes, wraparound, RF interference. These effects can impact any modality, and you have to do careful quality assurance to make sure you have good data. If you have one site that really doesn't do very much quality assurance, I mean, I haven't encountered sites like this, but it could exist. Or, some sites just do a lot more quality assurance with the scanner. You can have site differences or study differences in data quality that can propagate into your machine learning analyses. Okay, so at the group level or the population level, you can also have various confounds, and those are usually will depend on the focus of the study. So in some cases, age could be a real confound if you're looking at disease progression during periods of brain development, like in adolescence, or in older aging. Age can be a confound to looking at disease in those groups, for example. Sex, the example that I've given here, would be if you're trying to study individuals with a psychiatric disorder, frequently they have comorbid addiction problems, but there is a difference in the prevalence of addiction disorders between men and women. And so that could be a confound in your study as well. Motion, I mentioned, is always a confound. Younger and older participants are known to exhibit higher levels of motion than adults. In addition, patients with disease, like Alzheimer's or children with ADHD, exhibit higher levels of motion than their relative controls. And then factors like blood pressure, breathing rate, breathing depth, those can all cause changes in fMRI contrast and SNR, and those will be confounds if you're trying to study diseases like COPD or heart conditions or patients that have high BMI, for example. And then finally, to make these big data sets, you really do have to have multi-site initiatives to collect data in 10,000 kids or 100,000 people. You may also be motivated to combine independent data sets collected at different sites, even though they were not from the outset linked together. And when you do that, you introduce batch effects or site effects, scanner effects, whatever you want to call them, but it basically arises from having a data set collected at one site with a particular scanner manufacturer that may be different than the data collected at the other sites. You may also have differences in the populations that are imaged at a site in terms of the prevalence within the group of the different disorders that you might be interested in, or the numbers of healthy controls and so on. Variability across sites or batch effects can present confounds due to different scanners, data acquisition sequences, study protocols, stimulus delivery systems, behavioral and task paradigms, just a whole slew of things. Scanner effects, though, are a huge source of variability in multi-site data. Perhaps the one thing that you really do have to think about quite a lot because it's such a big source of variability. And, scanner manufacturer and field strength, differences in hardware and software, even upgrades within the same scanner can cause large variation. Daily fluctuations in scanner performance are also important. And these are compounded in multi-site longitudinal studies where you not only have inter-scanner variability, but you have a lot of within-scanner variation where you may upgrade the scanner over the course of a long data collection period for longitudinal studies. Okay, so how do we address these confounds? There are just as many ways of addressing confounds as there are confounds. So, I'm not going to be able to talk about all of the ways that you might be able to address confounds and harmonize multi-site data, but I'll touch on some of the ones that I'm most familiar with and that are actually commonly used. So the first thing you can do is try to harmonize your data across sites in advance of the data collection. And so this is what the HCP lifespan studies do, UK Biobank, ABCD, so on. In this case, you may choose to only allow sites to participate in the study if they're collecting data from a 3T Siemens scanner, right? So you keep the scanner type, the scanner manufacturer the same, you keep the imaging acquisition parameters the same or at least as close to each other as possible. You may have a scanner QA program based on phantoms or traveling subjects. Other things that you can harmonize are implementation, protocol implementation, non-imaging data collection, and then finally, if you go to all this trouble to harmonize the scanners and the acquisitions, you should also harmonize the data pre-processing, importantly, and the quality assurance, and that's critical for minimizing confounds. You can introduce just as much variability across sites by using different pre-processing streams as you can by having different scanners, I think. I don't know that for sure, but probably. So even after careful data pre-processing and harmonized data collection, you still have to even go above and beyond that and do this, further statistical harmonization. And those methods play a critical role, whether for multi-site studies with harmonization across sites at the data collection stage, or for combining independent data sets. So harmonization methods, though, all have a trade-off between removing unwanted variation associated with scanner and batch effects while keeping relevant biological information. Confounds are generally addressed at all levels of analysis. So it kind of starts, again, at the first subject level analysis. So for fMRI, confounds are usually included in that subject level GLM. So in addition to the task regressor that you might have in your general linear model, you also include temporal courses of whatever confounds you're going to model. In this case, it's fairly common to include motion time courses from re-registering those images when the subjects have moved. But you can also include global signals, CSF signals, and other derivatives from physiological measures and so on. And so by including these regressors in the GLM, you then estimate your brain map or your task activation map, statistically controlling for some of those confounding effects. So that's just a simple GLM approach. Confounds can also be modeled at the subject level for fMRI via data-driven independent component analysis. And then you use ICA, you apply ICA to the fMRI time series, you get a set of components and their associated time courses. So these are spatial maps and time courses of the signals that mix together to generate your bold fMRI data. You look at those spatial maps and time courses and you label them as noise, crap, garbage, or good stuff we want to keep. And then you take the bad stuff that you've labeled those components as bad, and you remove that from the fMRI data to get a clean fMRI time series. There are several advantages to this technique, and it's, I think, becoming fairly standard now to do ICA-based denoising of fMRI data, whether it's task or resting state. It makes no assumptions about the time courses or spatial patterns of the nuisance effects, can identify head and physiological motion, scanner artifacts, multiband artifacts, etc. And there are automated tools for ICA denoising of fMRI data. So tools like Fix and ICA Aroma do a really good job of cleaning up some of this garbage in the data. If you don't use an automated tool, the downside is that you have to label the components yourself. And that requires a certain familiarity with what ICA spatial maps and time courses of these effects look like. So, the automated tools are nice. Okay, so at the group level, then that was at the subject level for fMRI data. You include motion time courses and things like that in your GLM. At the group level, this is where you include subject effects like age and sex. You might include site variables to model confounds that occur, across the data set. So in this first case, You have a design matrix that has a part of the design matrix that is encoding the things you care about, like group differences or group membership. And then you have a part of the design matrix that are all of these regressors related to age, sex, and other confounds. And so you estimate the things you care about, statistically controlling for the things that you don't care about. You can also take a bit more of an aggressive approach and actually denoise or deconfound all the brain features by regressing out confounds and nuisance effects, and then taking the residuals as a clean data set. So in this case, the design matrix just includes maybe some summary measures of motion, even at the group level. So you have to do it at the subject level with the time courses, at the group level with some summary measure motion, summary measures of motion. Say that 10 times fast. And then things like age and sex and interactions and so on. These are just really two simple, but possibly the most common approaches for harmonization in the neuroimaging literature. Okay. Here also you would include covariates related to site or encoding the different sites if you have multi-site data, and that implements a basic harmonization of data across sites. That does assume a constant effect for each site, and it's not a very realistic model of site effects. However, in some big neuroimaging studies, it is a tractable way to do that. So I'm going to highlight one study here published by Smith. This was a big GWAS study using the UK Biobank. It's imaging GWAS, where they looked at data from more than 33,000 individuals from the UK Biobank study. These individuals all had data for 6 imaging modalities, and they used that data to compute almost 4,000 image-derived phenotypes. So volume of amygdala, connectivity between 2 brain areas, so on. They split the data into discovery and replication samples, and then separately for each, in the discovery sample, for each variable of interest, they did quantile normalization, and then they did denoising of almost 600 different confounds. So lots of confounds. Again, remember, with such large sample sizes, you're just sensitive to everything. So there are a lot of confounds that you need to consider. The reason I picked this paper was because they have this, it's not really a companion paper, but they have this paper that describes confound modeling in the UK Biobank. And this is a nice resource because it goes through all of these different confounds that you may need to think about in big neuroimaging. So some of the confounds they removed included things like age, sex, and head size. So those were subject-specific confounds. But they also had scanner and acquisition parameters and related confounds, 48 head motion variables, table position variables, and nonlinear and interaction terms. So quite a vast set of confounds there. Deconfounding of each variable of interest was done simultaneously via a single regression for each image-derived phenotype using all confounds together. I think you may guess that that probably removes some signal of interest too. But large sample sizes, imaging GWAS. So this is how they handled it. Another popular approach for deconfounding or harmonization, however you want to think about it, is the combat approach that was adopted from the genetics literature. It's a popular batch effect correction method used in genomics that's been adapted for correcting batch or site effects in multi-site imaging data. It's a GLM-based empirical Bayesian method for adjusting for batch effects, so to remove those site scanner effects while preserving biological associations. It's been adapted for multi-site diffusion MRI data, structural MRI data, and functional MRI data. For each site, a site-specific scaling factor is calculated, resulting in a model that adjusts for additive and multiplicative effects. And this technique seems to work quite well to reduce site variability. It's been further modified to harmonize data in the presence of nonlinear trends in the Pomponio paper, specifically age, nonlinear age effects. It's been used to estimate and correct for additive and multiplicative scanner effects, accounting for within-subject correlations in longitudinal studies, where each person has multiple imaging time points. It's also been used to model covariances. This method here by Chen is called CovBat, or correcting covariance batch effects, removes side effects in mean, variance, and covariance. And they showed that machine learning methods are unable to distinguish scanner manufacturer. In other words, which scanner did the data set come from, while retaining accurate prediction of disease group after their harmonization method was applied. Watchinger, I hope I'm saying that right here, showed empirical evidence for the presence of confounding biases that are passed on to machine learning models trained on pooled data. They did something called the Name That Dataset experiment that showed that scans can be correctly assigned to their respective data set with more than 70% accuracy using a random forest classifier, where prediction accuracy close to random chance of around 7% would be expected in the absence of dataset bias. They also showed that all the harmonization methods they tested, from simple z-scoring, which I didn't really talk about, to regression with site variables, that classification accuracy was reduced. So they all effectively removed some of that site variation propagating into the classification. However, all the methods also remove biological variation assessed in the study, and they suggest that you need to add supervision to the process by including variables that you want to keep in the data, like age and sex. And they also modified COMBAT to include additional variables reflecting effects that are desirable to remove, and they found that principal components computed across all the imaging features on the whole data set to capture generic variation, not specific to a single brain imaging measure, work the best. I think PCA seems like a good approach because site and scanner effects will be large sources of variation across the pooled data that PCA could pick up very easily.
And if I may stop actually, if you could go back, because there's so much just might be difficult to find later. So Chen's work, which looked at, it was scanner effects, right?
Side effects.
Side effects.
Yeah, which are mostly they consider as scanner effects, or they call them batch effects. The scanner effects contribute the most, I think, to site variation, but there are other things that can be called.
But at least,
yeah.
But on this work, it basically was saying that they were not really important in the sense, right, for the...
well, just the way that they evaluated their method, they looked, they showed that without anything, you could very accurately assign a data set to which scanner it was collected from. So
it was significant for classification, but for the overall goal or diagnosis, the information was preserved regardless. Right? That's what you noted? Or
yes, it is. With their technique.
Have I read it correct? I mean,
with their technique, yes, they were able to preserve some of the biological information that they cared about, but some of the other techniques that they tested were not. They were effective for removing those site influences or scanner influences, but they also did remove biological information too. So I think their method did a bit better of a job by including these PCA variables in there. They were able to develop a method that worked a bit better at removing those effects while preserving the biological information. Okay, so my lab has done a lot of work with ICA and data fusion. trying to develop these methods for data denoising. ICA and multimodal data fusion are unsupervised learning methods. I described this already for fMRI that can be used to separate signals of interest from noise or artifacts like scanner and side effects in multi-subject data sets. In this case, the subject series or the collection of brain maps from all your subjects for a modality or maybe multiple modalities are fed into the ICA or data fusion, which decomposes the data set into spatial patterns and a set of subject loadings on each pattern. So in contrast to the application of ICA to fMRI, where you get spatial maps and time courses, here you get spatial maps and subject loadings, right? And then you can, you have this same issue. You have a set of components in the subject loadings, and you have to label the components as noise or signals of interest. But once you label your components, the noise components, you can do deconfounding using either the resulting single or multimodal spatial maps, or you can use the subject loadings. And so I think ICA-based methods have a lot of potential because you can discover the problems in your data set. You don't have to make assumptions about what is contaminating your data set. You can look at it in a data-driven way. So we published this paper, my postdoc at the time, Hwanji Lee, in 2020, where we did multimodal data fusion of data combined across 6 different studies. And these studies had data collection periods that spanned a TIM trio upgrade. So this is a major hardware and software upgrade to a Siemens 3T scanner. We identified several components where the subject loadings were strongly associated with software OS, which reflected pre and post TIM. In this case, these are pre-TIM software versions, this is post-TIM, and that were also associated with study, with a study variable. We then used these patterns that we labeled as noise, and we denoised our data. And we found that this really removed a lot of these pre and post TIM or scanner effects from the data. So we broke our healthy controls into 2 groups, pre-TIM and post-TIM. And we did a group difference, we assessed the group differences. In this case, I'm showing gray matter density, or gray matter volume, and we found a lot of group differences in the non-denoised data. These are just healthy people. There shouldn't be any group differences. So we tested GLM regression, we tested single modality ICA, and then we also have our method, and we found that our method really did reduce those scanner effects considerably, a bit better than some of these other methods. Hwanji is doing further work to develop ICA-based approaches to take into account mixed components that might reflect correlations between site and scanner effects and effects of interest. So here we use a conservative approach. We only removed components with loadings that only related to the scanner and study variables, and not the variables that we cared about related to cannabis use. So that's a conservative approach. But she's doing work to try to use a what she called a dual projection approach for correcting the loadings from the mixed components to reflect only nuisance effects by projecting out the signal-related information. And then the corrected components, plus the components that are identified as only being related to site and scanner variables, can be removed from the data to generate a clean data set. I will say that if you were going to use one of these approaches, really, or any denoising approach for multi-site data with a machine learning method, involves cross-validation, for example, and splitting your data into training and test data sets. If you apply the deconfounding method, whatever it is, to the whole data set without separating train and test data first, that breaks the independence of the train and test data and that can lead to incorrect conclusions, right? So there are a couple of very nice papers that talk about this issue in detail and for cross-validation. Okay, so the very last slide is I'm going to show you some newer techniques for harmonization and deconfounding. Deep learning methods now are, this is sort of a really hot area of research, I think, for addressing this problem. So these are just a few publications. I could certainly spend an entire week probably talking about all the different ways that deep learning are being used here. Bento et al. Have a nice review of deep learning in large multi-site structural MRI imaging data sets that was published in Frontiers in Neuroinformatics in 2022 that discusses batch effects in the context of deep learning using pooled multi-site data. Okay, so there are a lot of different methods. There may be some limitations to the methods. You might need paired or traveling subjects data for training for each site. It may be difficult to validate the harmonized images that are generated from deep learning methods. These methods are also data hungry and difficult to train, and that has so far led to many of these methods being implemented in 2D or on patchwise bases. So we're not necessarily denoising the full 3D volume, just slices or something like that. The last paper by Dinsdale, they recently proposed a deep learning technique based on domain adaptation to harmonize MRI data for a given task using CNNs that could overcome some of these limitations. I thought this was a really interesting approach. Their method uses an iterative update approach with the goal of creating scanner invariant representations of the data that can be used to reconstruct the input images so that they contain minimal information about site or scanner while simultaneously maintaining performance on the main task of interest. So removal of scanner information from the feature space prevents that information from driving the prediction. So it seems to me like a denoising within the machinery itself. And they demonstrated their technique under different scenarios. I think it looks cool. That's it.